Good morning, my dear audience. So, I am Bishwajit Kundu back again with a new module called the protein structure function relationship. So, I will proceed with this. So, we will be talking about how important the structure of a protein is for it to function, and also the function of a protein, how important the function of a protein depends upon its structure. So, slight variation in structure of proteins actually results in tremendous variations in function. So, we will look at all those possibilities here with some very typical examples. But before we proceed, I would like to bring you back the same slide that I finished with in terms of protein structure to bring some continuity. So, here is the first slide, and there I told you that as soon as a protein which gets synthesized as a chain of amino acids from the ribosome inside a cell, this protein, depending upon what kind of amino acids are there, for example, they can be hydrophobic, they can be charged, they can be water loving, hydrophilic, and there are various other compositions possible, positively charged, negatively charged, aliphatic, aromatic, etc. So, depending on their independent propensities, as soon as they arrive inside the cell, they would tend to form a collapsed state by interacting with each other. So, I was giving you this example of this wire where the wire has components and each component has different possibilities or probabilities. So, this is like beads on a string, and then this component positively charged might interact with this component which is negatively charged. This being hydrophobic would like to interact with another hydrophobic, and so they will collapse immediately into a state which is a folded state. The folded state is defined by the amino acid sequence. So, the sequence of these actually define how the folded state will be assumed. So, if you have an unfolded protein, it actually immediately renders itself into a flexible intermediate, and that flexible intermediate undergoes some small rearrangements here and there, and finally results into a structurally native, what we call, and functionally uniform protein. And as I told you earlier, 90% of the time an unfolded protein, as soon as it gets synthesized, will ideally fold into this particular one given structure. However, under certain other conditions, this can fold into different structures as well, resulting into aggregate and causes various kinds of diseases. There are various models that has been proposed how a protein folds from this uniform three-dimensional flippy floppy structural noodle like structure to a compact folded structure to perform a particular function, but none of those have yet been proven absolutely, and all the models proposed appears to be correct in some or the other sense. So, proceeding further, I will now call upon some of the examples to make you understand how different and how important this protein folding is, or a structure of a protein is related to the function of the protein. You will be surprised to actually know that although in the previous slide here I have been saying that every time you have a particular sequence that will fold into a defined structure. Now, contrary, I can say a defined structure can be obtained if the sequence of amino acid remains constant. However, I will now throw at you some very interesting examples where the structure is defined definitely by the sequence, but the sequence may not be exactly same. Let us look at one such example first. So, the topic is proteins with different sequences may give rise to similar folded structure, and examples of that are, for example, the alpha lactalbumin. This is in association with an enzyme called the galactosyl transferase, transfers galactose to glucose and results in the formation of a beta 1,4 linkage, which results in the formation of another sugar called lactose. This is the primary sugar in milk, you know that. So alpha lactalbumin is a protein which is acted upon by galactosyl transferase, and this combination results in the transfer of galactose to glucose and helps in the formation of beta 1,4 linkage, resulting in the formation of lactose. Now, if you look at the structure of this is how it looks like. So, it is more like a lot of alpha helix starting from this blue end. So, you have some alpha helix, then goes up with some loops, forms another alpha helix, goes up with some loop and again forms alpha helix, and there is a small green colored beta sheet turn here, and then this is the structure. Now, interestingly, if you look at a very different protein called lysozyme. Now, what does this lysozyme do? It hydrolyzes the same beta 1,4 linkase that has been synthesized by the galactosyl transferase. Now, in one case this has been synthesized, in another case the lysozyme hydrolyzes or breaks this beta 1,4 glycosidic linkage in polysaccharides. And if you look at the structure of this lysozyme, you will be surprised to see, doesn't it look very similar to the structure that is up there? So, this structure of alpha lactalbumin, as you can see, is very similar to the structure of lysozyme. Now, these two structurally similar proteins, however, are different in their amino acid composition. So, if you look at the amino acid composition, each of these letters M, I think you may not be able to read it because it is too small from here to look, but I will read it for you. So, M, R, F, F, V, P, whatever, these are the amino acids one after the other, which constitute this structure of this protein. Similarly, these are amino acids. So, if you compare amino acid of lactalbumin with that of lysozyme 1:1, you will find that there are only about 35.3% identity, 58% similarity, and then there are some gaps and penalties which you do not have to worry about at this point of time. But given that there is only 35.5% identity, the structures being so similar, the functions being very different, actually tells a whole lot of information about structure function relationship. So, the function of one is synthesis, the function of another is breakage. The amino acid compositions of them are very different, but they fold into a structure which are very similar to each other. There are many more examples in literature. For example, there is one more here, which is the structural superposition of ovomucoid and CTF molecule. So, the ovomucoid is shown in green here, as you can see, and the CTF protein is shown in red. So, you can see a lot of superposition is happening somewhere between these two molecules. So, these and many more such examples are there. So, chymotrypsin and trypsin, they look very similar. I will give you that example in due course soon. So, given that the structure defines function, but then structurally similar proteins may have different functions. So, for example, here you have alpha lactalbumin and lysozyme. As I told you, I will be giving some more examples. For example, here we look at the proteinaceous food processing enzyme, that is the proteases. So, what are proteases? The enzymes that work on proteins. What do? If they work on proteins, they will break the proteins. So, when we intake proteins, whether you take pulses or fish or some other meat or whatever proteinaceous product we have, milk. So, those proteinaceous products, as soon as we ingest, as we intake, immediately undergoes processing by the help of enzymes called proteases, which ultimately releases the amino acids which are to be consumed and taken up by the system for processing or making its own proteins. Two very important proteinaceous food processing enzymes are the chymotrypsin and the trypsin. Let us look at them carefully. So, both chymotrypsin and trypsin are actually not present in the system as such. They are synthesized from their precursor forms. The precursor forms are called chymotrypsinogen or trypsinogen respectively. So, trypsinogen is a precursor form of trypsin, and chymotrypsinogen is a precursor form of chymotrypsin. So, both of them, because they have been named as gen, so they are called zymogen. So, these zymogens has to be converted into corresponding trypsin and chymotrypsin, which becomes the active proteases. So, there are some changes that needs to happen. That is what is more important in terms of structure, function, relationship of proteins. So, let us look at that. If you look at the functionality of these proteases, that is trypsin and chymotrypsin, so here there is an amino acid sequence which is a polypeptide. The amino acid starts from the N terminal saying alanine, serine, phenylalanine, proline, lysine, glycine and so on. And it has been shown by these arrows that chymotrypsin, which is a protease, that means it will break the protein, it works only when there are very heavy aromatic amino acids immediately after heavy aromatic amino acids. So, what are the heavy aromatic amino acids? If you recall, phenylalanine, tyrosine and tryptophan are the aromatic amino acids present in proteins. So, whenever in the sequence there is a phenylalanine or a tyrosine or a tryptophan, for example, here there are two arrows shown, one showing immediately after phenylalanine labeled in blue, and then a tryptophan labeled in blue. So, these are the places where chymotrypsin would work and break this bond between the phenylalanine and proline or tryptophan and aspartic acid here in this case. However, if you look at trypsin, so chymotrypsin is one enzyme, trypsin is another enzyme which actually cleaves or breaks immediately after the positively charged amino acids such as lysine and arginine. Remember, they have this ammonium group, here there is a group. So, these renders positive charge to these amino acids. So, whenever there is a lysine or arginine or a lysine again, so these are the places immediately after this bond gets broken by trypsin. So, trypsin acts on them and it cuts, it like cleaves those. Now, what happens in the digestive system? If you look at the digestive system, food is intaken, it goes through the esophagus to the stomach and finally reaches the duodenum, and then from there it finally reaches the large intestine and then gets eaten. So, at this place is the place where maximum digestion of the proteins actually take place. And what happens is, not only the liver supplies the bile which is important from the gallbladder, which is important, but also the pancreas secretes these two enzymes that I just now told you, trypsinogen and chymotrypsinogen, that is the inactive form of these enzymes, are secreted into the system. Once they are secreted into the system, this portion of the stomach is acidic, and this acidic environment causes a change in the structure of chymotrypsinogen and trypsinogen to form chymotrypsin and trypsin respectively, and those now starts working on the proteins that you have ingested through your food. The conversion, however, is also by cleavage, as I told you. So, conversion happens by, so chymotrypsinogen is produced in one form, one structure, it is not active. So, the structure in which it is produced inside the pancreas is inactive, but it is a structure, it is non-functional. But once it enters into this duodenum, the large intestine, immediately it gets converted into another structure, and that happens by two subsequent cleavages or so. So, chymotrypsinogen gets converted to chymotrypsin. The chain of the chymotrypsinogen, which is shown here, gets clipped in 4 places, and it releases 2 dipeptides, and it creates 2 breaks in the backbone, and that results need not have to go through all these distinct portions, but the end result is that you have an active chymotrypsin now, and this chymotrypsin now works in this fashion, that is, it cleaves immediately after phenylalanine or tryptophan or tyrosine, and then the protein as such will get cleaved. So, you see, one structure gets converted into another structure, one structure is inactive, but when it gets converted into another structure it becomes functional. So, the enzyme chymotrypsin is made up of 245 amino acids, and they are interrupted into 3 peptides. We call them one A, B, C. Now, if we go a little bit deeper into this chymotrypsin, why is it functional, how is it functional is something that we will learn. So, it becomes functional because there are 3 important amino acids, the histidine 57 which is shown here, the aspartic acid 102 which is shown here, and the serine 195 which is shown here. Remember, this is a polypeptide like this, which has just folded into some 3-dimensional structure, and then some amino acids here is histidine, another amino acid is aspartic acid, and the third amino acid is serine, which have come in close proximity because of this folding or the structure that it has taken. These 3 amino acids constitute a very important catalytic triad. So, when I say chymotrypsin works immediately after the heavy amino acid groups like phenylalanine, tryptophan and tyrosine, then these are the 3 amino acids, remember, histidine, aspartic acid and serine, histidine, aspartic acid and serine, that constitute triad or a triangle. It is called a catalytic triad. Let us look at how it works. So, normally the hydroxyl group of a serine, which is this one. So, you remember here is the serine looks like this. So, you have a hydroxyl right there, OH. So, this hydroxyl group of the serine is not acidic. So, in chymotrypsin only the serine 195, this 195 which was just a hydroxyl group and was not acidic, becomes acidic due to the presence of this histidine and aspartic acid in close proximity in the folded structure. So, when it folds, this serine comes in close proximity to the histidine and the aspartic acid, and then what happens, the specific site of chymotrypsin, that is this site, is a large space created in which these 3 amino acids are right there, and then a large hydrophobic group like the tryptophan, tyrosine and phenylalanine can come and sit there, and this is the mode of action. You need not have to worry about the exact details of this mode of action, but in general. So, you have D represents aspartic acid, H represents histidine and S represents serine. So, you have the DHS, aspartic acid, histidine and serine right there, and then what happens is this is the histidine and this is the serine. So, histidine and serine, this constitute the catalytic triad, and this is the incoming amino acid, that means the hydrophobic big amino acid that comes and sits here. Once it sits here, then it activates the serine, the hydroxyl of serine, now the hydroxyl, this OH now attacks the carboxylate of the incoming amino acid, and then through a set of these reactions which you need not have to worry at this point of time, but then it produces the first product, which is the cleavage of this product from the original polypeptide, and then it goes through another round of reaction through binding of water molecule, and that results in departure of the second product, and then this process continues. What is important for you to understand here is that there are 3 amino acids, aspartic acid, histidine and serine, the aspartic acid D, the histidine H and serine S. They come in close proximity because of the folding of the protein, and that particular structure results into a given or a particular function. So, you have to attain a particular structure to perform a particular function. Now, this is very, very important and interesting, because when I show you in the next slide what is more so important and interesting, you will become, it will become much more evident. But remember, these 3 amino acids have to come close enough for a function to happen. So, what is the entire all other polypeptides doing, the rest of the protein is doing? Actually, most of the cases, particularly in terms of enzymes, in most of the enzymes, the actual enzymatic action are performed by 2, 3 or 4 amino acids at the most. But then the rest of the structure is essentially to hold these 2, 3, 4 amino acids in place exactly in the 3-dimensional orientation, so that an incoming substrate can bind and the product can be released. So, it is like, I live in one house, although I need only a small place of a chair or a bed, but then this entire room is built around me, because it houses not only me, but the chair, but the table, but the fan or everything else that is required by a person to live in. So, the entire infrastructure, the pillars, the ACs or whatever is there, the fan, light, etc. is required for one person. Similarly, in the case of an enzyme, although it forms a structure, it has a large amount of amino acid all spread around, but it is only the 3 or 4 or 2 particular amino acids in the entire structure which will perform the function. But then those amino acids have to be brought in 3-dimensional space in a particular orientation for them to act. If there is any variation or deviation, then the protein becomes non-functional or inactive. Once this exciting trick of how a catalytic triad, for example, the histidine, the aspartic acid and serine, can cause breakage of proteins, long proteins. So, here is one protein which is an enzyme which has the aspartic acid, the histidine and a serine coming in close proximity in 3 dimension. This now acts on a whole gamut of different kinds of proteins and break it. Wherever there is a phenylalanine, tryptophan, tyrosine, it is going to break it. Now, this particular property of having 3 amino acids reaching a particular orientation to perform a particular function has evolved as a trick by which nature has repeated this trick in various other enzymes. For example, trypsin I have already told you. There are other proteins like subtilisin, thrombin and a very important brain enzyme called the acetylcholine esterase. Now, acetylcholine esterase is important for the recognition functions, because it helps in the nerve impulse transmission. So, nervous impulses transmission happens when you blink, when you hear, when I am moving my hands and legs, everything, everywhere the nerve impulse transmissions are happening. If somehow you block these nerve impulse transmissions, then you are essentially dead, because the heartbeat will also block, because there is nerve impulse transmission. So, you have to have these enzymes working all the time. So, you have trypsin, which definitely, as I told you in my previous slide, has this triad, that is the aspartic acid D, histidine H and serine S. Exactly similar nature. So, here this particular figure is of that of thrombin taken from this source. So, thrombin, subtilisin, acetylcholine esterase, all of them, these are 3 different, 4, 5 different enzymes altogether. They function at different places altogether, but then their mode of function or mode of action is the same. That is, they have the aspartic acid D, histidine H and serine S, and by a combination of this triad, the infamous triad, they break many more enzymes or proteins. They are called serine proteases, because serine or S is one of the central amino acids responsible for initiating the process of protein breakage by these enzymes or group of enzymes. So, they all have a reactive serine residue which is central to the catalytic mechanism. Now, I have highlighted or boldened this acetylcholine esterase here, because as I told you, this is important for nerve impulse transmissions. And after knowing that nature has utilized such a trick to actually accommodate a large number of enzymes having the same function, although structurally they may be slightly different, but they all hold these particular amino acids in position in such a way that they act as a catalytic triad. So, whether it is trypsin, the structure is different, subtilisin, the structure is different, thrombin, the structure is different, and acetylcholine esterase, the structure is different. So, all these 4 enzymes have different structures, but the function is same. What is the function? They will break other polypeptides or other proteins at positions wherever there is phenylalanine and tryptophan in the chain. So, as nature is very clever to have one trick discovered and then utilize that trick to produce a gamut of enzymes to function, similarly human are also very clever. They have now utilized this particular property itself to make something very important. For example, acetylcholine esterase inhibitors. What are they? So, you know nowadays organophosphates such as malathion or parathion, they are used as mosquito repellent, for example, in good night, Mortein or any other such thing. They affect nerve transmission in insects, but they do not affect nerve transmission in human. So, the structure of the human acetylcholine esterase is different than the structure of the acetylcholine esterase of mosquitoes and other insects. So, if the structure is different, their binding will be different, but the function is the same. So, what do? We use what is called as nerve gas, and this is how a nerve gas works. So, normally if you have this enzyme which is represented by this boxy appearance with some crevices and grooves, and then you have this serine which I have been referring to. So, this is acetylcholine esterase, and as I told you, acetylcholine esterase is having the same catalytic triad, that is serine, aspartic acid and histidine. So, here is the serine OH, and then acetylcholine, which is responsible or important for nerve impulse transmission to happen, comes and sits in this fashion. So, this is acetylcholine molecule, this entire thing, which sits in this fashion, forming the carboxylate group forms a bond with this hydroxyl group of serine, and then what happens is this acidic group is cleaved right here and it is produced like that, and the rest of the choline molecule goes out. So, acetylcholine gets cleaved in this fashion by acetylcholine esterase, and this particular entire function is important for the impulse transmissions to happen, nerve impulse transmissions to happen. So, what people have done, because now we have understood the trick of the nature, so what people have done is exactly this. So, this is what is called as nerve agent or nerve gas, which are organophosphates or malathions or parathions, etc. Now, what do? They come and just sit right here in the portion where serine would have interacted with their acetylcholine, and then this reaction does not happen. It blocks this reaction to happen, resulting in non-cleavage of acetylcholine into acetic acid and choline, and therefore, nerve impulse transmission does not happen. Therefore, it is a blockage of nerve impulse transmissions, and therefore, the mosquito or any other insect dies because their functionality is paralyzed. So, this is what nature throws at us in terms of structure of an enzyme and the function, and we utilize that structure and function information to our benefit in this fashion. So, this is bewildering science, this is what biology throws us, and this is what is exciting for us to study more and more about these enzymes. Another very important factor, you might have heard about this disease called sickle cell anemia. So, what happens is normally the red blood cells are spherical or disc like shape when these cells contains an enzyme called hemoglobin. Now, remember hemoglobin, I told you in my previous class that hemoglobin consists of 4 chains of polypeptides which come together to form a quaternary structure and holds an iron containing portion called the heme at the center, and that iron is responsible for carrying oxygen in the bloodstream. So, these are the red blood cells. These red blood cell consists of these proteins. This protein has a heme has an iron, and iron carries the oxygen. So, it binds to oxygen, and then this entire this red blood cell moves around in the blood vessels and then carries oxygen all through. So, that is what happens, we have understood that from nature. Now, if you look at any one of these polypeptide chains, whether it is this yellow colored A chain or the blue colored B chain. So, there are 2 A chains and 2 B chains. So, this is a quaternary structure, 4 different quaternary structure where 4 polypeptides have come together. Now, each one of these polypeptide has something called a glutamic acid at the 6th position. So, if you look at the chain of this hemoglobin, the chain A, that is this yellow colored. So, there are 1, 2, 3, 4 amino acids, the 6th one is a glutamic acid. And what happens is, if somehow by genetic mutation, by some other method, I do not want to name because that is a portion that you will learn somewhere else, this glutamic acid gets converted, can be genetic, gets converted into valine as is shown in this figure. So, glutamic acid is represented in yellow, it gets converted into valine, the green one. Then this chain, so look at this structure function relationship, the amino acid composition determines the structure. So, as soon as glutamic acid is there, it is fine, hemoglobin is working fine, but as soon as this glutamic acid gets converted into valine, then the structure changes. It no longer is capable of forming this beautifully evolved globular structure. Now, what has happened? So, instead of a globular structure like normal hemoglobin which is shown here, now in the presence of just one single amino acid chain in the entire polypeptide at the 6th position, this has now started clumped hemoglobin and no longer functions independently in this fashion, and therefore, their oxygen carrying capacity also decreases. So, it is caused by a change of amino acid in the 6th position of beta globin. So, this is alpha globin, if I am correct, this is beta globin. So, there are 2 chains alpha and beta. So, beta is shown in blue. So, excuse me for that, this is shown in blue. So, this is beta chain, and a change in the glutamic acid to valine results in the beta chain to take a completely different conformation. So, the structure is different, the structure becomes different, and then rather than assembling into this fantastic globular structure, they now start clumping together. This mutation results in one array of aggregates of hemoglobin molecule as is shown here, and then that results in deformation of the red cells which are shown here. So, now the cells look like sickle. What is sickle? Sickle is something with which you cut grass, So, those are the sickles. So, they start looking like sickle, that is why it is called sickle celled, and then this cause clotting in the blood vessels and tissues because they are no longer moving freely because their shape is such that they can get clogged at different places in the arteries. So, you see how fantastically a structure predicts a function. Just the structural change happens because of mutation, and once a mutation happens, the function is completely gone. So, with that, I would like to acknowledge very gracefully all those who have contributed to the different pictures, to the different animations from where I have actually taken these slides, which is purely for academic and teaching purposes. Thank you very much for listening to me carefully.